Good morning, girls. How are you all? Okay, we continue with our topic antibiotics. We were on this topic antibiotics. Antibiotics are of two types. Antibiotics are of two types, bactericidal and bacteristatic. This we did last time. Cidal, like I told you, cidal means killing and static means stable. So bactericidal, these are the antibiotics which are used to kill the microorganisms. And bacteriostatic, these antibiotics, using these antibiotics, the microorganisms will not grow further. They will not grow further. Like last time I told you, when the microorganisms will grow in a body and reach to some number, then only the body will show the symptoms of that particular disease. And if they will not grow to that number, then they will remain in a body, but body will not show the symptoms of that disease. So these kind of antibiotics are called bacteristatic. Examples of bactericidal are penicillin. The examples of these are penicillin and afloxacin. Afloxacin. Oflaxacin. Afloxacin. Bacteristatic. The examples of bacteristatic antibiotics are erythromycin, tetracycline, and chloramphenicol. Chloramphenicol. So these are some examples of bacteristatic antibiotics. We can also classify the antibiotics as narrow spectrum antibiotics, broad spectrum antibiotics, and limited spectrum antibiotics. Now what are yes girls, continue with the next part. We were on narrow spectrum antibiotics. Narrow spectrum antibiotics. These are those antibiotics which are effective against gram positive or gram negative bacteria. Now what are gram positive or gram negative bacteria is? Bacteria are causing the diseases, and these bacteria are of two types, gram positive and gram negative. So those antibiotics which are effective against either gram positive or gram negative bacteria, these are narrow spectrum. Their spectrum is narrow. These are narrow spectrum antibiotics. An example is penicillin G. Penicillin G. Now come to the second type, broad spectrum antibiotics. As it is clear from the name broad, their spectrum is broad. So they either kill or inhibit wide range of gram positive as well as gram negative bacteria. So bacteria, they cause the disease either by killing or inhibiting. Either the bacteria are killed by these medicines or inhibit, their growth will be inhibited. So these kind of antibiotics are called broad spectrum antibiotics. Example, amoxicillin, ampicillin and chlorampinicol. These are the examples of broad spectrum antibiotics. The next category is limited spectrum antibiotics. These are effective against a single disease or organisms. Now, one thing more about chloramphenicol medicine. Since it is broad spectrum, so it is effective against gram positive as well as gram negative bacteria. That means for number of diseases, one medicine can be used. So this is given in case of typhoid, dysentery, acute fever, pneumonia, and some urinary infections. So this is about the different type of antibiotics. Moving on to the next part, anti-fertility drugs. The last type. We know the problem because of the increase in the population. So to control population, that is childbirth, the drugs which are used, these are called anti-fertility drugs. So actually the birth control pills, they come under anti-fertility drugs. And these birth control pills, they actually have a mixture of synthetic estrogen and progesterone derivatives. Estrogen and progesterone, these are the female sex hormones, and these medicines, they suppress ovulation. Example of anti-fertility drugs are norethindrone, ethyl estradiol, which is also called novistrol, So These drugs are anti-fertility drugs which are actually controlling the childbirth. So this is our first topic that we have covered, the different type of the drugs. Now one thing more is left in the drugs, that how these drugs actually work. Drug action. What is the meaning of drug action? That is, how do drugs interact with targets? Drugs interact with targets. Targets, I told you, are biomolecules. Drugs targets are the biomolecules in our bodies. So this is drug target interaction. That is when drugs and biomolecules, they interact, then what will happen? Biomolecules are the targets. And we know they perform different functions in our body, like proteins are of different types. One example of proteins I'm taking is enzymes. And I think most of you know, but enzymes are the biochemical catalyst. They catalyze the biochemical reactions. Another example of proteins is receptors. What's the function of receptors? They are involved in communication. Our body communication system works because of the receptors. That, for example, if we touch something hot, immediately you withdraw your hand. So who tells to withdraw the hand? That is, that communication through the hand goes to the brain, and brain tells us to withdraw our hand. This communication system works through receptors. Then another example I'm taking of biomolecules is nucleic acids. Nucleic acids, they have genetic information. The information that transfers from one generation to the other is through nucleic acids. After nucleic acids, another example is lipid and carbohydrates. Their function is to form structural part of cell. Cell is made up of lipids and carbohydrates. So biomolecules, some examples of biomolecules I've given to you with their functions. Now, first example I'm taking is enzymes, to understand the interaction between drug and a target. Enzymes as drug targets. When enzymes will be the target of the drugs that we will take. Now first I'm coming to the function of enzymes, how they work. Enzymes, they have active sites on their surface to hold the substrate. Enzymes have active sites to hold substrate. Molecules for chemical reaction. Now to understand this, I am drawing a diagram. Just see. For example, this is an enzyme. And this is an active site on the surface of an enzyme. This is a substrate. or you can understand that these are just like two reactants. So substrate will fit into this active site, and because of this, a complex between substrate and enzyme will form. Substrate and enzyme will form. And substrate combines with this enzyme through some forces. These forces between the two can be ionic, or these can be the chemical bonds like the hydrogen bonding, van der Waals forces, or dipole-dipole interaction. So substrate links with enzymes through these kind of bonds. And after this, the enzyme will provide a required functional group to react with a substrate. And because of this enzyme and substrate complex, the chemical reaction will take place in a body. So the chemical reactions in a body takes place by the reaction of enzyme and substrate. When these two are linking, then only the chemical reaction will take place in a body. Now, let's say this kind of interaction is not required by a body. This chemical reaction is not required by a body because this is causing some kind of a disease or problem. Like for example, allergy is caused in a body because of this enzyme and substrate reaction. Now, for this allergy, we'll take a drug. To get rid of this problem, we are taking a drug, medicine we are taking. What that medicine, what that drug will do, it will either, two things are there. It will either compete with the substrate. Like for example, this is enzyme, this is substrate, and this is the drug that we have taken. There will be a competition between the two, between the drug and a substrate, that who will link with this active site. So here to the active site, if drug is effective, it will compete over substrate and will link to this active site. It will not let the substrate to link. Instead, the drug will link here at the active site. And because of this, now the enzyme, this is enzyme and substrate link is not there. So that reaction will not take place. And that reaction, I said, for example, was causing allergy. Since that reaction is not taking place, that means there will not be any allergy. That means the drug that we have taken is effective. These kind of drugs that we use, which are competing with substrate, are called competitive inhibitors. These are called competitive inhibitors. Now, sometimes the drugs, enzyme, this is substrate. The drug will not compete with a substrate. Instead, it will link at some other site in an enzyme. Not at this active site, it will link at some other site and change the shape of enzyme at the active site. Since this shape substrate cannot fit into this shape active site, so substrate is not fitting here. Enzyme and substrate, again there will not be any complex formation. So that reaction will not take place. So if the drug will link at some other site in enzyme, then these kind of drugs are known as, these sites, first of all, these sites are known as allosteric sites. These are called allosteric sites, and these kind of drugs are not competing with substrate. So these are called non-competitive inhibitors. Non-competitive inhibitors. I hope this will be clear to you. Then second example I'm taking for this drug target interaction is of receptors. Receptors as drug targets in the next video. Yes, girls. Second example of drug target interaction is receptors as drug targets. When receptors are the targets of the drugs. Receptors we have done already are proteins. And these kind of receptors are actually embedded in the cell membrane in such a way that the small part which has active site is projected out of the surface of the cell membrane. For example, this is a cell membrane that I'm drawing. This is the outer layer. And in this cell membrane here, receptors are embedded in such a way that small part which has active site that is protruding out, that is little outside. Now, how do receptors transfer the message to the cell? How they transfer the message to the cell? At these active sites, there will be some chemicals which will act as messengers. Messengers means these chemicals, they have some message. That message these chemical messengers will give to receptor when they bind here at this active site. And that message from the receptors will go to the neurons, from one neuron to the other neuron, or from neuron to the muscles. The communication system will work like this. Suppose this chemical which has a message in it, this chemical messenger, this kind of the communication is not required by a body. Then we are taking the drugs to get rid of this message. These drugs, what they will do now, again, they will not let that chemical messenger to give the message to the receptor. Instead, they will link at this active site. When they link with this active site, not this chemical messenger, what will link here is a drug. And drug will block this active site. So now when this is blocked, chemical messenger cannot link here and this message cannot be given to the body. It will not pass it to the neurons. Now sometimes what happens is, the chemical is not there at this active site. It has to give a message to the body. That message is required by the body, but that chemical messenger is not there. So for this, the drugs are taken. These drugs will do the function of this chemical messenger. The drugs will link here and these drugs will give the message to the receptor, and hence this way that function will go on in a body. That message will be given to a body. If the drugs, they bind to the receptor site and will stop the function of this chemical messenger, inhibits the natural function of this chemical messenger, then these kind of the drugs are known as antagonists. These drugs are used to block that function. Now sometimes, like I told you, that chemical messenger is not there to give the message to the receptor. Then we take the drugs, and drugs are doing the function of this chemical messenger. They are Mimic means they are copying the message of that chemical messenger. Then these kind of drugs are known as agonists, and these are known as agonists. These are the drugs that copy the natural messenger by activating the receptor. So these kind of drugs are known as agonists. These are useful when there is lack of natural chemical messenger in the body. And girls, with this this topic is complete. Drugs, different type of the drugs, and the interaction of drug with the target means with the biomolecules. Thank you. Moving on to the second topic of this chapter after drugs. It's a very easy topic. Chemicals in food. Which kind of chemicals we can add to food? First of all, these chemicals are added. Chemicals, they are added to food for preserving the food, for their preservation. Sometimes the chemicals are added to enhance the appeal of the food, to enhancing their appeal, that after using these chemicals the food looks good, so you will like to eat that food. Then adding nutritive value. For adding nutritive value, some chemicals are added to the food. Then fourth one is artificial sweetening agents. Instead of the sugar, the table sugar sucralose, we add artificial sweetening agents to give a sweet taste to the food. And from chemicals in food, we just have to cover this part, artificial sweetening agents. So this is our next topic, artificial sweetening agents. Natural sweeteners like sucrose, the table sugar, it adds to calorie intake, and therefore many people prefer to use artificial sweeteners. Example of artificial sweetening agents. First example is saccharin. This is the first popular artificial sweetening agent. This is 550 times more sweet as compared to the cane sugar, and it is excreted from the body in urine as it is unchanged. Its use is of great value for diabetic people and the people who need to control intake of calories. Second example is aspartame. Most successful and widely used artificial sweetener. This is most successful and widely used artificial sweetener. This is 100 times as sweet as cane sugar, but its use is limited only to the cold foods because it is unstable at cooking temperature. Third example is elitane. It's high potency sweetener. It's very, very sweet. So even if we'll take one drop of this, then the food will become very, very sweet. High potency sweetener, but more stable than aspartame. But the control of sweetness of food is very, very difficult using it, since it is high potency sweetener. Next example is sucralose. It tastes just like sugar and stable at cooking temperature and does not provide any calories also. So the better one is the sucralose. So this is a topic, artificial sweetening agents. First example is saccharin, second is aspartame, third is allitame, and the fourth one is sucralose. One more topic I'm covering up with this thing, very small topic, that is food preservatives. Food preservatives they are used to prevent the spoilage of the food. Food spoils due to microbial growth. And examples of food preservatives are table salt, sugar, vegetable oils, and chemicals like sodium benzoate, and salts of sorbic acid and propanoic acid. Sorbic acid and propanoic acid salts. These are food preservatives. I hope, girls, this topic is clear to you. And after this, approximately half of the chapter is covered with this thing. And you can ask your doubts on the WhatsApp only for half an hour. I'm giving the time to you. It's not that the whole day you will be sitting and writing the messages on the WhatsApp group. Since this chapter is very easy, go through this, and any doubts you have, you can write on the WhatsApp number in the next half an hour. Otherwise, in the next class also you can ask your doubts. Thank you so much, girls.